Welcome to AP Biology. Today we're going to talk about external signals at the checkpoints. So growth factors might be an external signal that will say, go, let's do cell division. So here's the growth factor attaching to its receptor, which will change the shape of the inside, the cytoplasmic domain of the receptor, which will then cause some kind of signal transduction pathway that helps the cell to get through some point in the cell cycle. So that's a go. And then other things are a stop. So density-dependent inhibition means if a cell is surrounded by other cells that are like it, they will stop dividing. Cancer cells won't do this. So here are cells in a petri dish or a culture dish, and they are all surrounded by other cells, and they don't divide. But if you scrape a few of them off, sort of like if you got a tissue wound, the cells would do more mitosis. What you can see here is the very much most outer layer of skin, the epidermis layer, and it's the cells here at the bottom layer that divide. And if you have a small wound, these cells will start migrating toward each other, and once they hit into each other, they'll stop migrating and they'll do mitosis until they fill this in, and then they'll do mitosis on the way up. One other thing to notice is that it's just this bottom layer that divides, and that is called anchorage dependence. So anchorage dependence is the idea that most cells need something to be attached to underneath. So for example, this cell is connected to adhesive molecules to the culture dish. So that cell can grow, it can do mitosis. This cell just in soft agar will usually just die. It'll do apoptosis and die. And that is very similar to these. So this basement membrane is what the lower layer of the epidermis is bound to. And these are the ones that do mitosis, and the upper layers don't. Cancer cells, however, will grow kind of anywhere. So cancer cells have a loss of cell cycle controls. They are told go, and they are not told stop. So first, let's do a few definitions. A tumor is a mass of abnormal cells within otherwise normal tissue. A benign tumor is one that's not cancerous. So that's, if there's a good tumor, that's the good tumor, depending kind of on where it is, but it's not cancerous. And then a malignant tumor means cancerous. So this is tissue, this is cells that can invade surrounding tissues. When they metastasize, that means that they're going to different parts of the body. So what you see here is a tumor. This can be reached with a scalpel, and so the doctor could remove the tissue, check the surrounding parts that were removed, and if the margin is clear, then you're in pretty good shape. And they probably will still do radiation and chemotherapy, but if you can catch a tumor in an early stage like that, that's excellent. Once it starts getting bigger, parts of it can break off, and that's what this means. When they are exported to other parts of the body. So they'll move through lymph vessels, which are sort of like blood vessels, but they have an end to them. And they just really, their job, a lymph vessel's job, is just to bring tissue fluid back toward the blood. Or they could move through blood vessels. And when they do, they can end up in different parts of the body. So a tumor from the breast might end up in the bone or the lung or the brain. I'm not really sure where they go to, but they can go to other tissues. And once that happens, you have a very different treatment protocol because the cancer cells are throughout the whole body. And it's not just one tumor that can be surgically removed, it's a lot of them. And so the therapy will be different. Angiogenesis means the growth of new blood vessels to the tumor. So if the tumor has released growth factors for blood vessels to grow toward the tumor and feed the tumor, then that's also not a good thing. Because if they have blood vessels, they will get oxygen and nutrients. And if they don't, then they'll be able to, it'll be a little bit more limited because they won't be able to grow as quickly because they won't have the oxygen and nutrients that they need. Okay, so how do cancer cells form in the first place? They don't respond normally. They do not respond normally to the body's control mechanisms. So it's sort of like being told to grow all the time and not to be told when to stop. So first of all, growth factors might be a problem. So for normal cells, if they have growth factors in petri dishes, then they will divide. And if they don't have growth factors, they will generally not divide. Cancer cells, though, will divide whether they have growth factors or not. So these are cancer cells, and whether they have growth factors or not, they're still going to divide. It doesn't really matter. So why is that? How come cancer cells will still grow even without growth factors. And so there can be several reasons for it. So here's an example of a cell that might be cancerous, and it might not need growth factors because it might produce its own growth factors. So you might not need to give it any in culture because it can just make them itself. Or it could instead have a mutation on the cytoplasmic domain of that receptor, and so it starts the signal transduction cascade even without a growth factor there. Or the receptor might be just fine and only respond when there's an actual growth factor from another cell. But it could be somebody in the signal transduction cascade that's mutated. So you remember how DNA makes RNA, and RNA, messenger RNA, will make proteins, right? And so if there's a mutation in the DNA, the particular gene that codes for, let's say, this protein, and it's always active, that mutation possibly makes it always active. It doesn't really matter what happens upstream. If it's always active, it's going to cause this whole transduction cascade. So that might be, any of those might be a reason for not needing growth factors for cancer cells to still continuously divide, whether even without growth factors, because maybe they make their own, or maybe their signal transduction cascade is happening even without growth factors. Okay, here's an example that you might see in an AP Bio test. So they might give you this diagram, and you don't have to memorize RAS or GTP or RAF, but you want to be able to look at the diagram and understand what's happening in it. So there are growth factors, and here they are, and they attach to the receptor, and then the receptor will change shape, which will activate, it'll be in the active position, it will activate RAS, which is interacting with GTP, so it probably is a G protein coupled receptor, I guess. Anyway, that will activate RAF. And then RAF causes some signal transduction cascade that causes cell division. Maybe it makes the cell produce a particular cyclin, for example. So let's say there's a mutation that makes the growth factor receptors always turned on. So maybe whether there's growth factors here or not, maybe these receptors, this part of them, the cytoplasmic domain, maybe that's always turned on. If that's the case, are you more likely or less likely to get cancer? And the answer there is you are. That cell is more likely to become cancerous. It would still need other mutations to become cancerous, but that is one more step in the direction to becoming cancerous. Well, what happens if the growth factor receptor is always off? Let's say this part can never activate. Well, that would make this cell less likely to become cancerous. It might have other problems, but cancer is not one of them. What if RAS is always turned on? So if RAS is always turned on, let's change colors here. If RAS is always turned on, RAS, that's this one. If RAS is always turned on, it really doesn't matter what happens previously. And so we're taking each of these one at a time, right? So if wipe the whole slate clean, and if RAS is always turned on, the cell would be more likely to become cancerous because it would activate RAF, which would then activate whatever is happening here. If RAS is turned off, that's the opposite. You would not be able to have the rest of this. Okay, what if RAF is always turned on? That's this one. So it doesn't matter what happened here. If RAF is always turned on, then you are more likely to have cell division and more likely to have cancer. And then the other way, if RAF is always turned off, then that's not working, and you are probably less likely to have cancer in this cell. Okay, so other things that can affect whether a cell does too much cell division. If a cell isn't told to stop, so the last slide we were talking about was the cell being told to go even when it shouldn't. And this one is the cell not being told to stop. So here are some normal cells in a culture dish, and they need to do mitosis. But once they're connected to each other, this cell is going to have other cells nearby it, and it'll have receptors on it, and so will the other ones. And they'll say, oh my God, you're a cell just like me. We are so in sync. Oops, that's the wrong way. We don't really need to divide because we're the same kind of cell. So that's called contact inhibition. If you lose contact inhibition, then here are some transformed cells. In this case, when you're talking about cancer, transformed cells means that they've become cancerous. So these cancerous cells will divide, but they don't just make a single layer. They just keep dividing and dividing and dividing because they don't know how to stop. They don't have contact inhibition. So they don't recognize each other. So these cells are like, there's nobody around me. I'm here all by myself. I guess I'll divide. They also lose anchorage dependence. So usually cells need to have something under them, some kind of basement membrane or adhesive molecules to attach to in order for them to divide. And if they're in just soft gel, they'll usually just die instead. But cancer cells won't. They will just keep dividing, which is why a cell can break off of, let's say, a tumor in the lung and move to the bone and still grow. Okay, so here's an example of loss of contact inhibition, maybe. Some of them are, some of them aren't. So here's what a dividing cell might look like. And this is supposed to be the cell membrane. And so the things sticking out here are proteins. And so if a growth factor comes over here and attaches to the receptor, that turns it on, which makes it change shape, which then turns on the next protein, which then activates the next molecule, which then activates the next one, which attaches to the next one, called CAM in this case, which attaches to RAS, like we saw a little bit ago, which then causes some other stuff to happen, which eventually maybe makes a cyclin for cell division to happen. Contact inhibition, here's what that looks like. So here is a cell membrane for one cell. So here's a cell, that's what's inside. Here's another cell, and here's its cell membrane. And then this is the space between the cells. So let's say a growth factor attaches here, and then that changes the shape of R, which then activates 1, which then activates 2, which then attaches to 3. 3 can't attach to CAM now. Well, that's because CAM is attaching to the CAM of the other cell, and that changes the shape of CAM. So now it interacts with M instead. And when that happens, 3 can't turn it on, so it doesn't interact with RAS. So cell division does not occur. So this is normal. This is what a normal dividing cell would look like, and this is what a normal cell would look like when it's in contact with another cell. So now let's look at the following mutations. Let's say that R is always turned on. So if R is always turned on, and I let's just forget about whether it's attached to CAM or not, whether there's another cell here or not. I just want to know if this particular mutation occurs, is that one step toward the cell becoming cancerous or not? So if R is always turned on, then it doesn't really matter if there's a growth factor. It's always going to activate 1, which will activate 2, yada, yada. So you are more likely for the cell to become cancerous. I know that if there's CAM to CAM binding, then that will stop the whole thing. But it is a bad thing if R is always turned on. It is just one more step toward a cancerous cell. Okay, let's say instead that 2 is always turned on. 2, that's this one. If 2 is always turned on, then that's going to interact with 3, which interacts with CAM, which turns on RAS, which causes cell division. That is going to increase your chances of cancer. I know you still have an out, right? I know M and CAM can save the day and stop cell division, but that's still a bad mutation, right? 2 always turned on is more likely to cause cell division than if it were a normal one. Okay, let's say instead that M is always attached to CAM. Well, oops, here's M over here. If M is always attached to CAM, that actually stops cell division. So that would make it less likely that the cell would become cancerous. Okay, let's say instead that RAS is always turned on. RAS. If RAS is always turned on, you are going to get cell division. And now it doesn't really matter if CAM is here or not. So that makes you more likely, makes this cell more likely to become cancerous. And that's actually a really, really bad one, because it doesn't matter if you have a growth factor saying go, and it doesn't matter if you have another cell saying stop. If that RAS is always on and always causing cell division, that's a really bad mutation, because you've lost both the stop, the contact inhibition, and you've lost the kind of caring whether you're told go. So that's a really, really bad one. How about if 1 is always inactivated? If 1 is always inactivated, then you're not starting all this stuff, right? If you're not moving forward, so that would be less likely to become cancerous, right? That means that growth factors could not start the whole process off. And then finally, if 3 is always attached to CAM, I have too much stuff on my slide here. Okay, if 3, whoops. Okay, let's say 3 is always attached to CAM. Well, that's not a good one, because that means that it doesn't matter if there's another CAM here, because M can't interact with it because 3 is always attached to it, which probably means that RAS can interact, which means that you're going to have cell division. So that is also a very bad one, just like this one. Okay, a few more things that go wrong when you have cancer. So another one is telomerase can always be turned on. So that's an enzyme that makes your telomeres get longer. So with your DNA, during synthesis, your DNA splits, not the whole thing, but parts of it, right? And so you have an enzyme called primase, which we'll go over this in another unit, but primase will make primers at either end, and then you will kind of make your DNA copy. And so I drew this kind of twisted ladder shaped, and I didn't do that with these. But anyway, bear with me. These are two DNA molecules. Then you'll come in and you'll do that for your whole chromosome, all of your chromosomes on both sides. You know, you'll use this side to copy another and that side to copy the other. Anyway, we will do a whole unit on that. But the primers get broken down toward the end, and then your DNA polymerase comes along and it will finish producing the rest of this, but it can't do it over here because it can't recognize that side of the DNA. It's a good story for another day. Anyway, the point is your DNA gets shorter. And look at this. This is an aging cell. And see the telomeres here are getting shorter. The whole point of telomeres is that they are just this DNA that doesn't really have any genes in it. It's just there because the chromosomes keep getting shorter. And so you want to have like a little bit of DNA that like doesn't do anything, so that it's sort of okay that every time you copy your DNA that you get it a little bit shorter. So when you get really old, your chromosomes are much smaller. So that's not a great thing. But when you have a baby, when you make your gametes, you have this enzyme called telomerase, which makes the telomeres back to where they should be, nice and long, so that when you have a baby, the baby starts life with nice long telomeres. So someone like me might be like, oh, coolio, wouldn't it be really nice to have some telomerase so I could grow some telomeres so I'm not so old? But that's not a good thing because telomerase is common in cancer cells. So it's one thing that makes them immortal. They can grow, copy themselves forever and ever and ever. Okay, cancer cells also fail to undergo apoptosis. So a normal cell, when it gets a bunch of DNA damage, will say, ooh, I have all this damage, I should die. That will save the rest of the body. Cancer cell, though, doesn't do that. Even though it has mutations, it just keeps on dividing.